Audio check. Okay, looks like we're good here. All right, let's get into chapter 2, chromosomes and cell reproduction. Be sure with this chapter, this is where we're going to start getting some more challenging material, although most of this will be a review for everybody. The core principles that we're going to learn in this chapter are absolutely essential. I'll show you towards the end what you need to understand inside and out to be able to answer the questions on the exam. Be sure that you answer the questions in the back of the chapter for this chapter. Very, very important. All right, this is a fluorescent microscopy image. So fluorescent microscopy is like regular microscopy, but it uses fluorescent dyes and then shines either laser light or UV light on the specimen. So it goes on one wavelength, and then the fluorophore, which is the dye that's on the cell, refracts the light. So it gets sent back to your eye at a different wavelength. That's the color that you see. And it's got a red stain for the cell membrane. It's got a blue stain for probably the DNA there. It's a typical dappy blue stain. And then the green represents the spindle apparatus, which is a network of microtubules that actually move the chromosomes. So, when we learn mitosis and meiosis in Bio 1, we learn all the prophase, metaphase, anaphase, and we're going to review that today. But that's not really what's most important to us. What's most important to us is how the chromosomes move and how they segregate. So we'll see that a little bit. So we're going to talk about also prokaryotes and eukaryotes, just comparing those two cell types. We'll do cell reproduction and also sexual reproduction as well. All right, prokaryotes and eukaryotes. Basically, prokaryotes are bacteria, and eukaryotes are everything else. All right, so plant cells, animal cells, fungal cells, everything. And they're much more complex. So if you compare the two, eukaryotes have membrane-bound organelles, and this gives way to higher complexity because now you have compartmentalization. And when you have compartmentalization, you can do more specific functions, right? Like a double membrane mitochondria can do a more specific function like aerobic metabolism. When you look at the genetics between prokaryotes and eukaryotes usually just have one chromosome, and it's usually circular. And eukaryotes have multiple chromosomes, and they're linear. The third major difference is size. Prokaryotes are much smaller than eukaryotes. In fact, if you look at a eukaryote, and you can see all those mitochondria in there, the yellow structures, a prokaryote is typically about the size of those mitochondria. So, you could fit hundreds and thousands of prokaryotes inside of a eukaryote. All right, yeah, so prokaryotes are unicellular, no membrane-bound organelles, no nucleus. Instead, they have a region where the single chromosome is all bundled up called a nucleoid. And it's not very organized. It's just kind of scrunched up in there in a particular region of the prokaryotic cell called the nucleoid. Whereas in eukaryotes, it's more organized. It has proteins that help package it together. Two subtypes of prokaryotes include archaea and eubacteria. Archaea, which kind of correlates to ancient, means bacteria that have been on the earth for millions and millions of years. So they've been around since the earth was a very, very different environment, right? When it was volcanic and acidic, things like that. So you see these bacteria living in places like, the boiling springs of Colorado and near hydrothermal vents or acidic lakes. Eubacteria is basically all of the other bacteria, pretty much the common bacteria that we're familiar with today. Archaea bacteria are more similar to eukaryotes than eubacteria is. All right, eukaryotic cells, unicellular and multicellular, and has membrane-bound organelles. It has a membrane-bound nucleus. You have the genetics are segregated away in the nucleus. And the DNA is, because you have so much of it typically, you have more than one chromosome, it gets packaged by these proteins called histones. This right here makes a unit called a nucleosome. But you can see it like wraps around a group of these 8 proteins in there. There's 8 proteins, 8 histones, and there's a linker right here. That's what you're looking at. But this enables the DNA to get fit inside the nucleus. All right, with several characteristics that bacteria and archaea have in common, and that distinguish them from eukaryotes. Give you a couple seconds there to maybe think about that. Bacteria and archaea are both prokaryotic, right? So that's what they have in common. And what makes them different is that bacteria and archaea have no nucleus. They only have a single circular chromosome. And it's usually much less DNA, a smaller amount of DNA. So how do prokaryotes, how do they divide, right? So it's a really simple form of division, unlike the organized step-by-step processes that go on for eukaryotes. Prokaryotes just undergo like a splitting called binary fission. But the most important event is that the chromosome gets copied. After the copying of the chromosome, then the bacteria just needs to split in half. The cell membrane pinches in and it splits in half. So 2 things to note about the replication of the chromosome is one, that on the chromosome you have a region called an origin of replication, which is where the copying begins. This is where the DNA polymerase enzyme is going to bind to the chromosome and then start building a new piece of DNA based on the code that's found in that chromosome. Now, what's involved in that DNA polymerase copying of DNA, there's another enzyme that also separates the DNA, right? Remember, DNA is double-stranded, and so you have to split it in 2, break the hydrogen bonds that connect the 2 strands of DNA. And when you separate DNA, it's relatively unstable because you've broken those hydrogen bonds, right? It wants to go back together. So there are proteins called structural maintenance of chromosome complexes, or SMCs, in prokaryotes that prevent them from going back together and prevent them from tangling up. There's a similar protein complexes in eukaryotes that help DNA replication, called single-stranded binding proteins. Prokaryotes also, because this is a very simple process, they can copy very rapidly. They have a very high rate of replication. In 12 hours, you can have billions of bacteria that just in a single culture, because they replicate so quickly. All right, eukaryotic cell reproduction, which is what we're going to focus on in this chapter, is highly organized and in a step-by-step fashion. All right, we're going to talk about eukaryotic chromosomes in this chapter, homologous pairs, chromosome structure, the cell cycle, which is different than mitosis, right? Mitosis is just a component of the cell cycle. And then we're going to also talk about the consequences of the cell cycle. All right, so these are some of these concepts that you absolutely have to understand like 200%. And that concept is ploidy. All right, so ploidy is the numbers of copies of genomes that a cell has, right? So all of our cells in our body that make up like our tissues, our muscles, our brain, our skin, our liver. They're all diploid, right? They're abbreviated 2N because of that. N stands for ploidy. They have 2 copies of the genome. 1 copy you got from your mother, and 1 copy you got from your dad. Right? So 1 copy from mom, 1 copy from dad. We have 46 chromosomes in our cells. 23 of them are from mom, 23 of them are for dad. Now, if you have a cell that is haploid, which is sperm and egg, they only have 1 copy of the genome. Okay? So be sure you fully understand that. All right, look at this. So here is a karyotype, which is a spread of all the chromosomes in the body. So in order to generate a karyotype, typically what's done is a cell is brought up to a stage called metaphase. It's called a metaphase spread. That's because you can easily arrest the cell at metaphase. And also all the chromosomes exist in this condensed state. So, they're nice and organized. You have this centromere and the long and the short arm clearly visible. All right, so if you look, you've got 23 pairs of chromosomes, right? Let's look at chromosome 2. We got this wacky looking one and then this straight one. One of these you got from mom, and one of them you got from dad. And because they are both chromosome number 2, they are the diploid version of chromosome number 2, we say they are homologs. They are homologous chromosomes. And if you look along the chromosomes, if you sequence it, the sequence would be very similar between the 2 homologs. There would only be some subtle differences. And that's because you have the same genes on there. Like, and let's blow this up. Here are 2 homologs here. Like here is the gene for eye color. Here's the gene for hair color, and so on. All right. But between the homologs, you may have different versions of the gene, right? You may have different alleles. This is the gene for blue eyes, and this is the gene for green eyes, for example. Okay. So these two versions of a gene encode a different trait, right? So here's allele A and here's allele little a. And we use these upper and lowercase letters to designate different alleles. Okay, so if this was allele big A and this was allele big A, that would mean they have the same exact allele. They have the same DNA sequence. But because this is little a, that means there's a small difference in the DNA sequence there, but difference enough to give it a different trait. This would be a different eye color. This would be a different eye color. These are homologs, homologous pairs of chromosomes. All right, diploid cells have 2 chromosomes, 2 sets of chromosomes, 1 set of chromosome, 2 pairs of homologous chromosome. What do you think? All right, you are correct. 2 sets of chromosomes, right? It depends on the species. So chromosome number species determines chromosome number. And just because you have more chromosomes does not mean that you are higher order species. A potato, for example, has, I don't remember exactly, but I think 90 something chromosomes or something. It doesn't connote to intelligence or anything like that. So yeah, 2 sets of chromosomes. Diploid cells. Haploid cells have 1 set. Diploid are 2N, haploid are 1N. All right, some more terms. So when a cell is like doing its normal thing, right? It's just kind of living its life. It's doing its normal function. It's the genes are getting read, RNA is getting made, and protein is getting made. The DNA in the nucleus is very diffuse. Okay, but before the cell starts to divide, the DNA starts to condense, and you can actually see the chromosomes. And when you see the chromosomes, there's like an attachment point usually in the middle of the chromosome, but not always, called a centromere. And it's a place where the chromosome kind of bends in right here. So these would be the centromere here, centromere here, right up with these blue ones. This would be the centromeres up here. So it kind of invaginates in a little bit. And that's going to be the attachment point for that spindle apparatus that we looked at way over here, right? That green stuff, because it's going to be able to pull the chromosomes apart. And there are motor complexes near that spindle called kinetochores, which are really complex protein complexes, and they are going to help delineate, which cell the chromosome goes to, right? Does it go to this daughter cell or the other? The ends of chromosomes are protected. It's not like you have a cap of proteins on them. There's actually specific redundant sequences that occur at the end of chromosomes called telomeres. And that's because we're going to learn about this later on. When these chromosomes get copied, because of like the limitations of DNA polymerase, the very tips of the chromosomes will eventually degrade after successive rounds of replication. And so you have these protecting sequences, and there's enzymes that work with them to kind of replenish the telomeres. But this is something that's implicated in aging. As our cells generate over and over again, chromosomes actually get shorter, and that's been linked to aging. All right, so remember, if in the prokaryotic chromosome, that one circular chromosome, there was a single origin of replication to copy that whole chromosome. But with these linear eukaryotic chromosomes, you will have many origins of replication along the chromosome, multiple origins of replication. All right, so here we've got one chromosome, and here we've got a homologous pair of chromatids, but they still make up one chromosome, okay? This is where the nomenclature is absolutely crucial, that you start to understand this. And you need to understand it at the level that you can draw it from memory. That's how well you understand it, to be able to answer the questions that you're going to be presented with on the exam. All right, single chromosome because it's by itself, but when it's paired together at the centromere with its homologous pair, we now call them chromatids, and we consider this to be one chromosome still. Okay, you can see the kinetochore is in green, and the blue are the spindle, the microtubules, spindle microtubules that are going to attach to the centromere. And it can, now pull this homologous pair apart. So pulling apart this one chromosome into two sister chromatids. Okay. You see the telomeres, the sequences at the end, kind of keep the end stable. Okay, so now if you remember back in that figure, keep bouncing back to that. I'm sorry. If you look, like the centromeres are not the same in all the chromosomes. Most of them are they're in the middle, metacentric, but there's some that have like the centromeres on the edge, or halfway, three-quarter way point or something like that. It does vary, and there's different terminology. Metacentric is in the middle, right? Submetacentric is like that three-quarter way part. Acrocentric is really far along the chromosome, just so you have these little kind of buds that protrude from the top. And telocentric, you have only two arms, right? So it's a very distal, which means far, very distal extension of the centromere that only provides two arms. But typically, the centromeres are submetacentric and acrocentric, so that you have long and short arms, right? The long arms are abbreviated Q, and the short arms are abbreviated P. So here, these would be the P arms right here. These would be the Q arms. Okay. All right. So be sure you know those terms. That'll be on the test. I have one of those on there. All right. So knowing what you know about the kinetochore and what it does, what would happen? Chromosome didn't have one. What do you think would be the consequence of that? We didn't talk about much about what's going to happen during mitosis, but what do you think would happen if a chromosome lacked a kinetochore? So if it lacked a kinetochore, it wouldn't be able to attach to the microtubules. It wouldn't be able to get segregated correctly to the new daughter cell that's going to result from mitosis. And eventually it would just degrade because of that. And a daughter cell would be missing now a chromosome. This does happen. It forms some relatively common chromosomal mutations where one cell will maybe get an extra chromosome and one cell will lack a chromosome, things like that. We'll talk about those mutations. Okay, so we're not only going to talk about mitosis in this chapter, but also the cell cycle, right? So, a cell's life is defined by the stage of the cell cycle that it's in. And it's usually in the stage of interphase, right? Where it's just doing its day-to-day operation. It's not in an active state of division. So in interphase, right, it's an extended period between cell divisions. It's reading genes and making protein and sending nerve signals or making hormone or contracting, whatever the cell does. It includes these different growth phases. They're abbreviated G. They're also sometimes abbreviated or called gap, right? Gap phase. There's two of them, gap 1 and gap 2. And then there's a very important phase within interphase called S phase, which stands for synthesis, right? So that means all of the DNA is going to get copied. Now, if you're in S phase, that means the cell is committed to divide, right? Then there's this little tiny phase called M phase, which is active mitosis, all right? So here's the cell cycle. It's going in this direction. Life is going in this direction. Let's start here at G1. So gap 1. During this process, during this stage, the cell is growing. And this is really considered, of all of interphase, which is right here, this is the stage where the cell is just doing its normal life, okay? Now, alternatively to entering the G1S checkpoint and committing to division, some cells can actually enter a stage where they become quiescent, or this GO. And when cells become quiescent, well, they can remain there for life. Occasionally they can, sorry, one second. Derek, I'm doing something. You got to get out of here, bud. Close the door. No. Get out, please. Not right now, bud. Get it and be quiet and get out, okay? All right, so cells enter the G1S checkpoint, and they can, at that point, they're committed for division. Come on, Derek, don't do that again, okay, buddy? All right, sorry about that. All right, so what were we talking about? We were talking about cell quiescence. So occasionally those cells can come out of quiescence and then go back into G1, but it's rare. There are signaling pathways that mediate these processes. All right, so gap 1, the cell's growing, it's doing its function, and then all of a sudden it receives a signal, such as a growth hormone signaling signal to divide. All right, so what it does is it passes this checkpoint at that point. There are different proteins like cyclins and cyclin-dependent kinases that activate pathways that made this decision for the cell to divide. Once it's past the G1S checkpoint, it's committed. So the first thing the cell needs to do is duplicate all of those chromosomes. It's got 46 chromosomes in there, and in case of humans, and all those chromosomes need to be copied, right? So we've gone from now a diploid cell. We're still diploid, right? The terminology here can get a little confusing, but we're still diploid, but we have double the number of chromosomes, okay? So we have 92 chromosomes in there. So this phase is very important. All the DNA is getting duplicated. And then after all the chromosomes are copied, it enters G2, where the cell is getting ready for mitosis. And during this stage, like, all of the mitochondria are getting doubled, and the membranes that make up the endoplasmic reticulum are increasing in size, so that when it splits, the lysosomes are increasing. Everything that the cell's the two new cells are going to need is important. So at the G2M checkpoint, basically the cell is checking that all of the chromosomes have been copied correctly, because that's the most important part, right? The two new daughter cells need a correct copy of the blueprint, the genetic material. And if all the chromosomes have been copied correctly, the cell can now enter mitosis. And mitosis is active cellular division, where the chromosomes start to move correctly, and then eventually cytokinesis is where the cell splits. There's an additional what's called checkpoint that occurs. It's like the anaphase checkpoint during mitosis. It's checking the correct attachment of the spindle and stuff. Not as important as the G1S and G2M checkpoints, though. All right, and those checkpoints are key, like stages of the cell cycle, because once they go through that checkpoint, why do you think this stuff is so intricately studied? Well, mistakes in the cell cycle, problems with cell cycle, are heavily implicated in cancer, right? Cancer cells are cells that divide out of control, so they have problems with the cell cycle. All right. So I think I've covered all of this basically in that previous slide. I don't think I missed anything here. Biochemical, yeah. All right, mitosis is the separation of sister chromatids. Cytokinesis is the separation of cytoplasm. These are the stages of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. And you may have learned this different in different books. Sometimes there's a pre-prophase and there's a sub-metaphase. It's all the same stuff, man. This is the way this book does it. This is the stages we're going to learn. Again, this is not as crucial as looking at the chromosome number and the slides that are about to follow this section. All right, here are the stages of mitosis. Here's a regular interphase, which is actually not part of mitosis. So it's a little confusing there, but interphase, remember, is a separate stage of the cell cycle. And then prophase, prometaphase, telophase, anaphase, and metaphase. What's happening during these stages? Well, prophase, the DNA is condensing. Nuclear membranes breaking down. The spindle is starting to develop. The centrioles, right, which produce the spindle, are separating to different poles of the cell. All right, during prometaphase, the nuclear membrane is completely broken down. And then ultimately during metaphase, all of the chromosomes, and you can see they're all doubled here, right? Remember, there's the chromatids are connected together at the centromere, and they all start to line up in this area called the metaphase plate. All right, and here's the metaphase plate, metaphase. And then anaphase, the chromatids are pulled apart. And then during telophase is basically the reverse of prophase. The nuclear membrane reforms. The chromatids are really now termed chromosomes and whatnot. All right, let's see here. What is the correct order of stages in the cell cycle? Now, if you look at this list, they don't have them all. Like we're missing, G2 is not even in here. So that's okay. It's just what is the correct order of these stages that are given. So if you look, G1, S, prophase, metaphase, anaphase, that one looks correct, right? So this would be, if this was correct, it'd be G2 there. let's see if I'm right here. Yep.